My dear friends, once again I welcome you to my YouTube channel. Today we will be discussing learning about an important chapter in genetics in 12th standard, Unit-VII. Unit-VII is about principles of inheritance and variation. In this, one aspect is a law of dominance, incomplete dominance, codominance. Let us learn about these aspects. Mendel conducted the experiments with pea plant. He selected 7 traits, in each trait contrasting characters. Height is trait, characters are tall and dwarf. Monohybrid, dihybrid. With regard to monohybrid, he proposed a law of segregation, law of dominance. With regard to dihybrid cross, he proposed the law of independent assortment. Now, these are the experimental results in monohybrid cross. Trait is height, one variety tall, another variety dwarf. These were pure lines, homozygous, pure for that particular character, pure for tallness, pure for dwarfness. These are parents. He crossed the tall plant with the dwarf plant. The F1 generation, he got all tall plants. In F1 generation, first generation, he got all tall plants. He made self-pollination between the tall plants of the F1 generation. Self-pollination. So that gives rise to next generation, F2 generation, second filial generation. In second filial generation, he observed both tall plants and dwarf plants. With his mathematical background, he counted all the tall plants, all the dwarf plants, and ultimately came to the conclusion that the tall plants and dwarf plants are produced in 3:1 ratio. Now here, when tall plants are crossed with the dwarf, in F1 generation, tall plants appeared, only tall plants. That is, the tallness is dominant over dwarfness. Tall character is dominant, whereas the dwarf character is recessive. One is dominant, another is recessive. That's why it is called law of dominance. So again, this is another monohybrid test cross, parental, tall, dwarf, TT. Mendel just imagined that there should be some factor in the plant that dictates tallness. Capital T, small t, small t. Then when these plants produce the gametes, that means pollen grains and the ovules or eggs. Today we know that reduction division will take place. They will be haploid, gametes will be haploid. So the gametes of this plant will carry only T, whereas this small t. In F1 generation, they come together, they stand side by side, stay together for some time, capital T, small t. The phenotype is tall. For external appearance, the plant is tall, but from genotype point of view, it is heterozygous. One factor determines tall factor, another small, t small t. So both are tall, but this is homozygous, this is heterozygous. So this is Punnett square. The gametes, then all possible combinations were taken. Which gamete unites with the which we cannot tell. But all you have to take into account all the possibilities of the gametic unions. Phenotypic ratio 1:, phenotypic ratio 3:1, genotypic ratio 1:2:1. Then in some other experiments, taking other characters, even Mendel, after Mendel also some people conducted the experiments with other plants with other traits. For example, here you see one variety, pure line variety, red flowers, another with white, another white. Capital R genotype, small r here. That means we can say this is dominant character, this is recessive character. Red is dominant over white. We can say at least time being. Then in F1 generation, the phenotype is pink. Interestingly, he got the pink flowered plants. Pink color is an intermediate character between the red and white. It is neither pure red nor completely white. Pink, intermediate character. Then he proceeded with the as usual experiments, F2 generation. In F2 generation, he got like this, a red 1 ratio, pink 2, 1 white. That means phenotypic ratio red, pink, white 1:2:1. Similarly, genotypic ratio, this 1, this 2, this is 1, 1:2:1. Here there appears to be some dominance, but not complete dominance. In dominance, complete dominance, ratios are 3:1, 1:2:1. Phenotypic ratio 3:1, genotypic ratio 1:2. But in this case, incomplete dominance, both phenotypic and genotypic ratios are same, 1:2:1. So these were confirmed by Mendel, and the later scientists taking selecting other plants and other characters, they have the law of dominance is not always true, but sometimes it will be incomplete. Remember, in law of dominance, in F2 generation, phenotypic ratio is 3:1, genotypic ratio is 1:2:1. But in incomplete dominance, in F2 generation, I am speaking about F2 generation, both phenotypic and genotypic ratios are same, 1:2:1. Then codominance. While conducting experiments with some other plants, some other animals, they have observed a third type of dominance, that is called codominance. It is a type of intra-allelic interaction in which alleles of the same express equally in heterozygotes. Now, let us take dominance. One is completely dominant, another is recessive. That character doesn't appear in F1 generation. In incomplete dominance, dominance is there but incomplete. The pink colored plants, flowered plants appeared, incomplete. But here what happened? both the characters appear side by side in F1 generation. Both are dominant, both are recessive. In codominance, both the characters appear in F1 generation side by side. That means both the characters are dominant and at the same time recessive. Do not interfere with the expression of character of another. In law of in complete dominance, what is happening? Tallness is dominating over the recessive or dwarfness. In incomplete, dominance is there but incomplete. But here, one character does not interfere with the another character with regard to their expression in F1 generation. There is no blending of characters, means mixing of characters. They are free, they are independent, they are expressed at the same time, side by side. Skin color, cattle, human blood groups, these are the two best examples for codominance. This is a codominance in cattle, a red coat, white coat. They have crossed these red, white skin color. In F1, roan appeared. These roan colored means the skin will be with patches. Some patches are red, some patches are white, side by side. Both are expressed side by side. That means one character is not interfering with the another character. Then in F2 generation, again this 1:2, 2:1. One red, one white, two roan, three or one red. So in codominance, both the characters are expressed, both are dominant, both are recessive, both are expressed side by side. One doesn't interfere with the another. This is skin color. Again, representation, genotyping, capital R, small r, capital R, small r. These are roan with skin with mixed color. Codominance in human blood groups, this is very important, very likely to be asked in NEET. Inheritance of AB blood groups is also classical example of codominance. In blood groups are controlled by three alleles. What are those? A, B, O. Some are one allele, one gene is A, one gene is B, another is O. Now, A, B are codominant to each other. Suppose if they are together, A and B genes are together, both are expressed. O is recessive. O is recessive. Three alleles, remember, in blood groups with regard to dominance and recessive. Three alleles, A, B, O. A, B are codominant. A, B are codominant. Now, for example, let us take gene A. Suppose a person is with A gene. On the surface of the RBC, there will be antigen A. Such people are referred as A blood group people. Some people will have B gene, B antigen on the surface of their RBC, blood group, B blood group people. Some people O, no antigen, then they are O blood group. Now let us see genotypes. Suppose a person is with A blood group, then alleles he may have AO heterozygous, AA homozygous. Both the alleles are same here, homozygous, but here heterozygous. One allele is A, another is O. But even if O is there, he is referred as A group because this is recessive. This is dominant. Therefore, genotypes AO, AA. B blood group people, two types of genotypes. BO, BB. Here it is heterozygous, homozygous. Both the characters express the same. Both the genes express the same character. Here heterozygous. But even if O is there, because B is dominant over, he will be expressing the B antigen. So therefore he will be known as B group. Here both. Similarly, AB, there are some people with AB blood group. One allele will be A, another allele will be B. One A, B. They are known as AB. Suppose let us take another person, O blood group, OO. Both are recessive, no antigens. Therefore, O blood group genotype is O. Now, human blood groups. Let us take this. Parent 1, parent 2, offspring. Suppose father, mother, children. Blood group of offspring, children blood group. If A blood group father weds, marries, A blood group mother, woman, all the children will be A blood group. Father A group, mother A group, all the children will be A group. Then A group, B group, mother B group, father A group, mother B group, children AB blood group. So they get one allele from father, one allele from mother. And here AB, codominance, they are AB. And now similarly, let us take AI, 0. Suppose mother with O group. Father A, mother O. Children will be A group. Then similarly here, father B group, mother A, then they will be AB group. Reverse to this. Then father B group, mother also B group, children will be B group. Then father B group, mother O group, children will be B group. It is not difficult to understand. Then father O group, mother also O group, children, all the children will be O group. So this is how the alleles are inherited to the children when father, mother, their blood groups. According to their blood groups, we can tell what will be the blood group of the children or offsprings, whether male or female. So this is, suppose AB blood group father, AB blood group mother, both are AB group. Then gametes of father AB, gametes of mother AB, then Punnett square independent mixing. Then what will be the phenotype? Among the four children, one will be A group, two will be AB group, one will be B group. Genotypic ratio 1 to 1. So both the phenotypic ratio, genotypic ratio 1:2, 2:1. Now you can tell if both are AB blood group, then what will be the possibility? One child will be with A blood group, VG for blood group. Two children will be with AB blood group, and the fourth child will be with B blood group. This is how, what is the blood group of the parents, father, mother, then what will be the blood group of the children, we can determine, we can say. Another example of codominance is beta thalassemia minor involving mutant of hemoglobin B child. So this is another aspect. Let us not consider this at this stage. So thus we have discussed what is the dominance, law of dominance, law of incomplete dominance, red, pink, white, codominance, red, roan, white. Then codominance example, cattle, human blood groups. There will be three alleles, remember, A, B, O. A blood group people will display antigen on the surface of their RBC. B blood group will have the antigen B on their RBC. O blood group, no antigen. So, in this case, who will marry whom? What is the blood group of father? What is the blood group of mother? What will be the blood group of the children? This we have discussed. Very important. You are likely to get a question from this. I think you have followed. Thank you. Follow my lectures. Subscribe the channel. Like it.